So with that, let's get started. First, I want to introduce myself. I am Can. I am a senior PhD student in the Safari Research Group and a lecturer at ETH Zurich. My research interests lie in the intersection between bioinformatics and computer architecture. Specifically, I'm interested in designing algorithms and architectures to enable real-time analysis for genomic data, and also designing algorithms for genome editing and error correction and so on. You can get to know us, of course, and our research, our group using these websites, and contact me using these links over there. So these slides are already available on the course page. So what we're going to cover today is some basic introduction to genome analysis, to genomics. I'll give you some basic background, and then I'll show you some step-by-step how we perform or how we analyze genomic data, and then we'll show you what are the state-of-the-art algorithmic and hardware acceleration works for genomics. And we'll briefly touch on the future opportunities for analyzing the genomic data. So we need faster, scalable, and accurate genome analysis because it is important for many reasons. For example, it enables us to uncover and treat diseases that are linked to genomic variations. It also enables us to alter the genomes directly to solve fundamental challenges of life. It enables us to detect the pathogens living among us so that we can take an early action, for example, for rapid surveillance of disease outbreaks. And of course, there are many, many important applications based on the genomic analysis data. But then what is a genome, right? So a genome is what enables us to have the different traits, right? So different, perhaps, eye colors, different phenotypes, and so on. So the genome is, loosely speaking, especially at least for eukaryotic genomes, so these are the DNAs, bunch of DNAs or the chromosomes that are located inside your cell, specifically in your nucleus. And for human genome, we have 23, in most cases, we have 23 pairs of chromosomes, and these chromosomes contain certain nucleotide letters that look like this, right? So when you convert the DNA into a digital form, you'll see a bunch of ACTGs. So the next question is essentially how large is a genome, or maybe more specifically, how large is a human genome? So what you're seeing here is one of the beautiful buildings in Zurich, specifically located in Oerlikon. So this is Andreasturm. So this is about 100 meters long. So to give you some illustration on how large a genome is, so if you wanted to basically start from the beginning of the first chromosome and then write every letter on A4 page until you reach to the end of to the last chromosome for a single cell, and then you pile up all these papers on top of each other, what you'll get basically a paper that is as high as basically this Andreasturm. And if you are curious, I did the math here, it checks out. So since this is basically a very also important area that has many promising applications, people started looking at essentially the human genome and try to decipher the human genome. And this effort, although it started even a lot earlier, given the improvements in the technologies, many scientists started to basically to recover the human genome starting from 1990s. And then this took around 13 years, and it costed around $3 billion so that we could decipher 3 billion bases that we have in the human genome. And this is, of course, very important because we can now look at the human genome and then see what makes us human and what makes us different than other living organisms and other individuals. But this first human genome, human reference genome constructed was not fully complete, although it took 13 years and billions of dollars to generate it. As the sequencing technologies, the machines that we use to generate the genomic data, as they improve, and also as well as the algorithms improve, we could basically complete the gap that was previously in the initial version of the human reference genome. We could complete that, and so that we can finally have a near complete of this human reference genome. And this was so, let's say, important such that these 4 individuals that were behind completing the human reference genome were selected as one of the most influential people of 2022. So we've been talking about the human reference genome, so let's take a little bit closer look at the other species. So here what you're seeing is the illustration of a viral genome. It usually has around 5,000 bases in its genome. When we move a little bit further, what we see is a bacteria. This is specifically a E. coli bacteria, and in its genome it has around 5 million bases. So when we move a little bit further, we have the mammalians, specifically here the Homo sapiens, which or humans, which has around 3.2 billion bases in one of the pairs of their genome. And essentially, you might be seeing a trend here. Essentially, as the genome size increases, maybe the complexity or the intelligence increases, but this is not specifically the case, because as we move further, what we see is basically an onion, which has a lot larger genome than the humans, and we even have larger genomes. So basically, this tells us something, which is the genomic data. Yeah, go ahead.
Is that per cell? And
yes.
From cell to cell, do they differ, or are they?
This information is per cell. Actually, even more specifically, this is per haplotype. For example, the Homo sapiens, the mammalians are diploid, meaning we have actually one pair of essentially 2 copies of chromosomes. And when you just take the 1 copy and then look at a single cell, this is the amount of the bases that you're going to see. Actually, in total for 2 pairs, it's around 6.4 billion. But then the plant genomes are essentially a lot larger because they are not diploid, they are essentially polyploid, meaning they have actually multiple copies of the same chromosome. But here we're still looking at a single cell over there. Given your other question about whether it differs cell by cell, loosely speaking, they don't differ much. You hope that they don't differ actually much. If they differ too much, then this is likely that you're either copying something like for too much, which may cause perhaps a cancer, or you have a huge deletion basically, which can be another problem. So loosely speaking, all of the cells have around the same amount of bases for the same species. Okay, so let's move on then. And so basically DNA is usually referred to as the blueprint of life, right? And the reason for that is because it is used in the later step to create the functionality. And actually, I like to use this, although it is not perfectly accurate, I like to use the analogy of using source code and then building applications with it. And this goes by essentially imagine your DNA is simply a source code, right? DNA, source code has useful parts, but also it has useless parts. Maybe you have some comments or essentially some parts that will not be basically translated into a functionality later on. So this is the similar case in DNA, and this DNA is usually transcribed into RNA, and this is similar to basically translating your source code to an assembly code. So here the functional parts are taken out. Now you have the instructions over there so that you can then translate this RNA to protein so that it makes the functionality similar to how you execute basically these instructions on your microprocessor. And we look at the DNA because as I said, it is important also to understand the functionalities and so on, but it also enables us to compare the individuals to each other so that we can identify whether there is a particular DNA portion that makes us more prone to have a particular disease. And this is usually referred to as genome-wide association study. So usually what you have a control group, maybe people with a particular symptom, for example, a heart disease, and you have your control group without a certain symptom, and your cases with a certain symptom, maybe with heart disease. And what you do is essentially you compare the genomes of these individuals and then try to find whether there is a particular variation that we consistently see in our case group, but not in our control group, so that we can link this particular mutation to, for example, having a particular disease, right? So if you consistently see, for example, a particular mutation or single nucleotide polymorphism, or called SNP, in this particular group, we can say, oh, maybe when you have this SNP over there, you're going to likely to have a heart disease. So this is a statistical analysis that you may be doing. And what you can do, you can get your genome sequenced, and there are these open databases out there, so you can already identify your SNPs, and then you can query your SNPs in these databases to see whether these SNPs or these mutations mean anything, right? So you can check them out from these websites. So as we also covered earlier, there are also, of course, much larger variations, so structural variations that cause even more serious, perhaps diseases or other symptoms. And essentially, the applications are, I would say, are only limited by our imagination. So it's not just about linking the diseases to each other. So one application that I'm really interested is the genome editing. Today, what we can do is we can pinpoint a particular region in your genome, and we can perhaps alter that content in the genome, so that perhaps maybe you disable the functionality in your genome that makes you prone to a certain type of cancer, right? And of course, this is an important discovery, and the people were behind this idea, which is essentially showing that we can program this genome editing and then almost target anywhere in the genome to alter its content. So this was awarded by the Nobel Prize to these two individuals. We also want to do large-scale analysis, right? So we don't really necessarily look at a single individual. We want to analyze maybe the entire population quickly. So doing the genome analysis in a large scale is also very important. The genome analysis is also used for the rapid surveillance of disease outbreaks. For example, it was used to identify the Zika virus spread, also used for COVID, and also the Ebola surveillance and so on. Genome sequencing is also used for testing purposes. There are papers that show how to do that. And also, we can perform in-field portable genome analysis. So imagine you attach your drone with a sequencer, and then you let the drone run in a field, and then it does the analysis for you. Maybe in an agricultural environment, it continuously checks whether that farm is now contaminated with something dangerous, so that you can take an early action. And of course, we want to do also the genome sequencing and analysis in challenging environment, maybe in even the outer space, to see basically how certain organisms behave in the outer space. Maybe even in Mars, there are papers discussing this. Of course, DNA is also a very valuable asset to protect, right? This is a very sensitive information, very sensitive data. And this has become actually more important very recently, because I'm not sure if you heard of it, but there was this relatively recent leak. Actually, this happened last year in 2023, where people would usually send their genomic sample to this company that is called 23andMe. I'm not sure if they are under now or they are still surviving, but this was a huge leak. But then what happened? What was happening is that when you sent your sample, they would be analyzing your genome and then would be giving you some insights, right? But essentially, apparently they were keeping the data about you, and this data got leaked. So certain individuals now essentially have the very sensitive data of many individuals, many people. And this is essentially very important because so genomic data is very important, right? And one example of how this can be used against you is, imagine you want to get a health insurance, and then somehow your estimate is much higher than what you expected. So you don't understand, or you don't know why your estimate is higher, but perhaps the insurance company who secretly has the data about you maybe knows why, and that's why maybe they are giving you that code that you cannot understand why. So this is basically one example of how this can be used in a hostile way, right? And you don't want people to use it for such reasons. Another application is you can also shop with your DNA. So imagine you go into a grocery and then you're not sure whether you're allergic to a certain product. So what you do, perhaps you read the barcode of the product and then send it to the company that knows about you, knows about your genomic data, and it can quickly tell you whether you're allergic to it or not, right? So you can quickly get some answers about the product and about you and whether you really need to buy it or even maybe more insight. So you can see basically how this information can be used in the daily life. Furthermore, people also use DNA to do the computation. So, for example, we can now solve hard problems, even a traveling salesman problem, by using the DNA. And how people do it is essentially they encode the DNA in a way that this DNA will make the connection only if there's a certain path in the graph. And then what you do, you just let the DNA do its natural job. It makes the connections and so on. And after some time, since this is a huge, massively parallel computation, the compute unit, let's say, after some time, if there's such a path in your graph, it will essentially do the DNA connection. And then you can sequence and then see whether you have that answer or not. So you can do the computation even with the DNA now. So as I said, the applications are limitless. And even maybe there are more applications that we cannot think of now. So the other question is, how and where to enable this fast, accurate, cheap, and scalable genome analysis, right? And today, what we usually do is that we have these special purpose machines that we use for data generation, specifically to generate the genomic data. So they are essentially very fast because they are designed to generate the data at a very fast rate. But essentially what we're doing is we're trying to analyze this data using these general purpose machines for data analysis. So this machine, as you can see, it is pretty scared because it is slow, and it is scared because it doesn't know how to analyze this data in the best way, right? So that's why the data generation speed and the analysis needs do not necessarily always match with the hardware that we're using underneath it. And to resolve this problem, we're looking at essentially different architectures and constantly asking the question, given this particular data type and given the requirements of the application, what sort of architecture or sets of architectures we should use, right? So this can be GPUs or SSDs or in-memory computation and so on, basically the devices and the architectures that we've been covering so far in this course. And even maybe mobile solutions, right? You can connect this genome sequencer into your mobile phone and perform the analysis. So this means that it needs to be also energy efficient computation. And of course, to achieve this, as you've probably seen this figure so many times, we need to look at basically the entire stack of computer architecture to co-design the algorithm architecture and software altogether. So if you're interested in learning more on how we essentially accelerate the genome analysis, you can take a look at this paper. This is a review paper. It's an invited review paper that was presented last year and published and presented last year at DEC. This is also another work that shows how we, what are basically the current efforts to accelerate genome analysis. So now I'll quickly cover some step-by-step analysis of the genomic data. So in high level, we usually start with the sample collection. So this can be your blood, right, or your tissue, or basically any particular cell that might be coming from an organism. And then we sort of prepare it so that the library that we prepare that contains your genomic data can speak to the machine, which is the genome sequencing machine. And then this genome sequencing machine generates initially the raw sequencing data. Then we use this raw sequencing data to do the analysis. So here, most of the computational steps essentially are here, and we're mostly interested in these parts. And the common genome analysis pipeline includes multiple steps, and this starts with the genomic data generation. So what we initially have is essentially a biological molecule, which is maybe your DNA or RNA. which is not human readable, right? So we need to somehow convert this biological molecule into a human readable form. And to do so, we use these machines that are called high throughput sequencing machines that essentially generates fragments from this biological molecule and then converts them into the sequences of nucleotides that we call reads. There are certain challenges to this data analysis. One challenge is now once we convert this into digital form, their origins are lost. So we don't know where they are coming from now in your genome. The second challenge is these sequences may contain some variations or errors, so you need to deal with them. The third challenge is there is essentially a large volume of this data that we need to analyze and compare it against a large space of the reference genome. There are many sequencing technologies that we can use to generate this genomic data, and each of them, depending on the technology that they use underneath, they generate essentially different types of raw sequencing data. And we look at essentially these newer sequencing technologies and the opportunities related to them. I'll basically mainly focus on one particular sequencing technology, that is Nanopore sequencing technology, because we're essentially interested in this direction, as you'll see in the coming slides. So this is, in high level, this is how Nanopore sequencing works. So here you have this mini device, a very small device that is called a mini-ion sequencer, and this can literally fit in the palm of your hand. So this is a small device, and inside this there is this flow cell that is the consumable that you buy to generate your sequencing data. It has some essentially ASICs and sensors so that it can convert the raw data or the analog data to the digital format. But in the very low level, what it has is these bunch of tiny pores that we call nanopores. And these nanopores are actually the little devices that perform the sequencing. And as output, it will generate the electrical signal data. So, but of course, this is the current state of this nanopore sequencing technology. So we can now fit this entire and large-scale sequencer into a very small device. But it didn't start with this initially. So it started with an idea in the 1980s by David Deamer. And the idea was essentially, can we create a three-layer solution, or let's say, even to make it even simplify, can we create a two-layer solution? Here, maybe one layer is charged with particular ions, and the other one is charged with the other type of ions. For example, here, this part might be positively charged, and this part might be negatively charged. So David was thinking about this idea because probably he would know that the DNA is also negatively charged. And the question was, can we essentially move this DNA from one region to another region while we have a current flow between these two regions? And can the movement of this DNA disrupt the ionic current flow? And would this ionic current disruption be informative for us so that we can tell each base, each nucleotide in the DNA? So this was the idea, right? So we want to look at the ionic current disruptions, and then based on these disruptions, we want to tell each base in the DNA. So in terms of idea, it is brilliant, but it didn't work well back then. And the reason for that was, when you put the DNA over here and then you create the electrical charges according to, for example, this part is negatively charged and this part is positively charged, and then you want to let the DNA run from here through here, it essentially was moving too fast. So you could not, basically, you did not have the technology, the sampling technology, to sample the ionic current disruption for every base that is moving inside this narrow area. So there were two challenges. One, we needed to slow down this DNA somehow, and we needed to basically improve the technology so that we can increase our sampling rate. So as the time moved on, of course, this technology improved, and we could now essentially sequence every base, not just once, on average actually 10 times. so that we generate electrical signals 10 times for every nucleotide to increase your confidence. So this is nanopore sequencing, and due to its essential benefits, it's a widely used sequencing technology. It can produce long fragments of DNA up to 4 million genomic bases. It is portable, so you can carry it around and use it if you want. It is relatively cost-effective, and it provides some more unique features that I'll cover next. So this is basically how a portable nanopore sequencer looks, and inside it there are thousands, maybe a few hundreds to thousands of pores, tiny pores. And each of them is essentially contains a 3-layer solution, as I explained earlier, right? So we have negatively charged region, positively charged region, and in the middle we have a membrane where this nanopore is attached. And what is happening is that this motor protein, which was essentially very essential to slow down the speed of this DNA, it initially attaches to the DNA, and then it guides it towards this nanopore. Once this attachment happens, what happens is that this DNA literally, it first performs a essentially double strand break, and then a single strand is now moving from one region to the another region. And as it moves through, it generates electrical signals. We measure the ionic current changes or disruptions, and these electrical signals are generated at a certain throughput. And what we can do is that as this raw signal, electrical signal is generated, we can start analyzing these electrical signals. Even we can perform real-time analysis, meaning we can start analyzing them instantly as they are generated. And furthermore, we can even make some real-time decisions. For example, based on our real-time analysis, we may realize that whatever we're sequencing now is not useful for us. So we can tell the sequencer to stop sequencing that particular read, and so that it can move on to another DNA, so that we can utilize these consumables even better. So there are two main benefits to real-time analysis, to summarize. One is it will stop sequencing, it will reduce your sequencing time and cost. But the second, it will enable you to overlap the sequencing time, the analysis time with sequencing, so that you can reduce the latency. So this is essentially how we generate the genomic data. But initially, what we have is a raw sequencing data. So what we need to do, we either translate this raw sequencing data into the nucleotides in a human-readable form. This is called base-called sequencing data, using, for example, some neural networks. Or we can somehow process this data by reducing the noise inside it and then perform the analysis directly without base calling. So these are two traditional approaches. One, we translate, and then we have ATGCs, and then we perform our analysis. So this is usually more accurate because the translation removes almost all the noises in the electrical signal. And there are, of course, many tools that were developed that uses this base-called sequencing data. But this translation process is very costly, and it requires the use of power-hungry devices such as GPUs. There are more recent works that aim to directly analyze this raw sequencing data without the translation step. So they provide efficient analysis with better scalability and portability. And also raw signals retain more information than just individual bases. For example, you can infer the chemical structure of the DNA also from the electrical signal. So then the question is, do we need a translator to analyze the genomic data? And the answer for now is it depends on the application. But what you can do is you can process the electrical signal such that you recognize somehow the passing of nucleotides somehow by identifying the abrupt changes in the signal, so that you can assign or use the values that corresponds to sequencing of a certain DNA, and then perform the analysis using these values. So we are interested in this direction, and we essentially published several works towards this direction. For example, this work enables the analysis of raw sequencing data in real time without translation. This is a subsequent work that improves its accuracy and performance. We also want to identify the mutations in raw sequencing data without even base calling. So this means that we need to somehow align individual signals to our reference genome. So this is what row-align does. So it performs the alignment so that we can identify the mutations in the raw sequencing data without base calling. We also look at how we can make the base calling part more efficient, more accurate. So this is a framework that discusses many opportunities and optimization techniques to reduce the complexity of your deep neural networks that are mainly used in the base calling works. So we also look at how we can perform base calling using in-memory or using memory computation. As I said, the base calling heavily uses neural models. So this means that you have a bunch of, for example, multiply and accumulate operations. So what you can do is you can rely on certain analog computations to perform these multiplications and accumulations, and then so that you can do your neural net computations and perform the base calling. So this reduces the latency and also reduces the energy consumption by doing the computation in memory. There are also some errors in the sequencing data, so we use machine learning techniques to understand and correct these errors. So this is basically how you prepare, let's say, this sequencing data so that we can analyze it in the later step to identify the origin of sequencing data and also to identify certain mutations and variations. So we do this in a step that is called read mapping. Mainly, the aim of read mapping is to solve life's puzzle from the sequencing output. And we do this by comparing the sequences to each other in a practical way. And we need to do this quickly and accurately because otherwise it would not be practical given the large space of genomic data. So I like to use the puzzle analogy over here. So assume that your biological sequences, your reads, your DNA are maybe the pieces of a puzzle, right? So initially, you don't know where each piece belongs to. So what you can do, for example, you can compare these sequences to each other so that you have some sort of an idea what this particular genome looks like by comparing them to each other. Or you may have a template, right, that we call reference genome. As it also so happens in the puzzle solving case, you have a picture that you look at and then see, try to identify whether this region belongs to here or somewhere else, right? So this is how you would use the reference genome. But the idea is essentially to have an understanding of where these sequences are coming from and what are their exact differences to each other. And this is essential to understand the functionality of a sequence because it enables us to identify the mutations, diseases, and so on. So if you want to look more closely on how we perform the mapping or the puzzle solving of the reads, so this is how the DNA logically looks like. This is how physically it looks like. But initially, what we have is these bunch of reads. And our goal is to, given this reference genome, is to identify where each of these pieces are coming from, and also identify their exact alignment, exact changes compared to those regions. So this is basically called read mapping. And doing this practically is important because these reads are usually very short and you have millions of them. And then you want to, for example, look at the human genome that includes 3 billion characters. So you can imagine the size of the problem, or you don't even have to imagine. You can take a look at this illustration and then assume you want to do the mapping of a read to a reference genome. This is a single read to a reference genome in a naive way, meaning you perform your approximate string matching algorithm for every base of a read to every base, starting at every base in the reference genome. So if you do this, basically this is a long process. It requires a large number of approximate string matching computations, which eventually is not basically very practical to do so. So somehow we need to reduce this complexity, and then we do it basically by relying on some heuristics and also data structures to perform this similarity identification in a practical way. So imagine we have this reference genome. So this can be huge, right? Maybe 3 billion characters, and then you have your read. So what we initially do is that we store certain subsequences from a reference genome inside a data structure that we call hash table, or we refer to it as index as well, where when you see a certain subsequence, you store it as a key in the hash table, and then you store its position as a value. So meaning when you query this hash table with a subsequence, what you'll get as an answer is list of positions where this subsequence appear in the reference genome. Or if it doesn't appear, you'll get a no response. So we use this hash table so that, so yeah, this is what I just described. We use this hash table so that we can also extract some subsequences from the read and then quickly query these reads using this hash table so that we can identify some potential similar regions between a read and the reference genome. Then we will eventually have some multiple candidate regions, some regions that we think that this read might be coming from, but most of them will be false. So to identify these false regions, we can also perform some filtering or some other algorithms that are known as chaining so that we can remove some false matches. Then lastly, we perform our approximate string matching or the alignment step, which is a costly step, but our goal is to reduce the execution of this as much as possible after seeding and filtering. Then we perform this alignment so that we can identify the exact matches and exact differences between this read and the reference genome at the position where we think that this read belongs to. So this is basically the steps in read mapping to make the sequence comparison practical. We look at essentially how we can perform this seed matching or subsequence matching accurate to basically improve accuracy and the performance of read mapping. So if you're interested in this direction, you can take a look at this paper. But eventually what we see is that the dynamic programming computation inside the read mapping, which mainly is used in alignment and chaining, consists of almost the entire execution time of read mapping. So the rest of the steps, for example, the seeding, the filtering, and so on, they are not essentially causing a huge computational overhead, but the DP computations. So maybe one could look at how we can accelerate these steps. But second question is, do we really need to rely on also a single, yeah, go ahead. Nanopore sequencer does not give you a fixed length read. So the length of them will vary a lot. And I would say theoretically, there is no limitation to the read length that the Nanopore sequencing could sequence. But due to the imperfections in the library preparation step, for example, even you shake your library a little bit, some of the DNAs will get fragmented very quickly, so it will break them off. So due to these imperfections, and of course, as the DNA gets longer, there is a higher chance that it will get tangled. So if you disregard all of these imperfections, theoretically, in the theoretical side, there is no limit to the read length that Nanopore sequencer can read to.
What is the length of the subsequence?
So this is a good question because it depends on the application, and also it depends on the genome that you're using. For example, if your goal is to identify the exact position of the read, usually those subsequences are of length 16 to 21, 22, and so on. So there are some theoretical works behind it that shows like why this is a good number. But if your goal is to identify whether this read is coming from a particular genome, particular organism, you're not looking at basically what is its exact location, but whether it belongs to a certain genome, then you can increase that subsequence length a little bit more, maybe up to 50 to 60 bases. Given if the amount of information or level of information you want to get is coarser, let's say, so that you can tolerate maybe some caveats that is caused due to the exact matching of these subsequences, right? But then maybe in some other region, like maybe you have a pool of reads, maybe you have 100 reads, but maybe it's just enough for you, just it matches, like one of them matches, and then you identify. Are there more questions? Okay, then let's move on. So the question was, do we really need to use also a single reference genome, right? So this is what we showed earlier. This is maybe the image that you're looking at, you're taking a clue at, and these are the pieces, these are the fragments from your genomic data, right? So and their lengths are different given the technology and so on. So by looking at a single reference genome, what we do is we recover the origin locations, right? By looking at a single one of them. But the problem is, if your organism or if your sample that you're looking at is somewhat relatively too different than the reference genome that you're relying on, then this means that maybe this can be due to certain huge variations. Maybe there are some structural variations that you cannot recover. Then this means that you'll only map a few of these pieces, and then you'll miss mapping some of the other pieces that are too different than the reference genome that you're looking at. So this is called a reference bias, right? So you're looking at one image, and then you see that, oh, this is too different. Oh, maybe then these pieces are not coming from this organism. But this is not necessarily true. These reference genomes do not represent the entire humanity, but it's only a collection of certain individuals. So the idea, like one solution to it, okay, let's not use one reference genome, then let's use multiple of them. But this is not also very clever, right? Because then this means that you're scaling your problem n times worse. Maybe you're increasing your runtime also n times if you use n many reference genomes. So what is an idea then? The idea is to use graphs. And the insight behind it, in most cases, for most regions, the organisms from the same species, they will be highly similar to each other. So they'll only differ at particular locations. So then this means that you can eliminate this redundancy. You can efficiently store this redundancy using a graph structure, right? And you can still have, let's say, edges and nodes for the regions where maybe individuals tend to differ. So this is basically the idea behind using a graph to represent the reference genomes. And this is essentially a recent approach that the field is moving towards to. And we're also interested in these directions. And this is essentially how the genome graph would look like to represent many different variations that a single reference genome cannot represent. So this is the read mapping step. So the last is to utilize this information that we generate from the read mapping step, which could be maybe the positions that you identify, maybe the alignment information you identify, and then somehow use it in a useful manner. So there are many use cases. Maybe you can identify similarities between reads so that you can reconstruct the genome. You may want to look at, you may want to pile up basically these reads to each other and then take a consensus of the alignment information to identify the variations. Or you may want to identify the presence of the genomes in your sample so that maybe you can take an action according to it. So this is called metagenomics. The goal of metagenomics is to identify the organisms that you have in your sample, which are unknown initially. So these are a bunch of DNA that you don't know where they are coming from. And there are many steps to perform genome analysis. One is you extract the k-mers from the reads. Then what you do is, you use these large databases that contain the information from many species instead of maybe a single reference genome. And then you try to find basically the matching k-mers between this database and the reads that are coming from your sample. And then once you have these matching subsequences, as you also asked earlier, what you can do is that maybe you can confidently say, if this particular k-mer is matching to a certain region, certain genome, you are very confident that this read must be coming from that organism. So by doing so, you can count, perhaps take the relative abundance estimation of each organisms in your sample, and then you try to see whether you have a bunch of bacteria, viral genomes, or harmful pathogens growing in your sample. So I think this is a good time to take a break. So in the second half or almost the second half of this lecture. I will go slowly because I realized that actually I've been going a little bit fast, assuming that I wouldn't be able to finish all the content on time, but hopefully we'll be able to do it. So you're welcome, everyone. We're going to go slower. Okay, so in the almost second half, relatively second half of this lecture, we're going to cover the algorithmic and hardware acceleration works for accelerating genome analysis. But before that, we need to understand that there are significant barriers to achieve fast and efficient genome analysis. And one big reason is there is a significant performance gap between the data generation and data processing, as we covered before. And when we look at basically the throughput at each step, starting from the data generation, where you sequence the data and you generate your data, and then starting from the translation step, you move further in the genome analysis pipeline, you perform your read mapping and maybe variant calling. So when you look at basically the throughput at each step, you'll see there is a huge imbalance between steps. And this is because each step doesn't necessarily know what the prior or the next step is doing. So you don't necessarily pipeline them really well, or some steps really require you to fully finish the prior step so that you can start processing or performing the analysis in the later steps. So there is basically not a clear solution to streamline this nicely, starting from the data generation up until we get the answer that we want to get from the genome analysis. And yes, so we showed this slide before. One big reason is there's a huge mismatch between the data generator and the machine that we're using to analyze this data. The second, the other major issue that we're seeing is the data moments are expensive. That probably you already know from the previous lectures. But the second, we have many of them in genome analysis. So when you start from the sequencing machine, remember we're generating a large volume of data that we need to analyze. And this data usually needs to sit at the SSD, at the disk, and then it needs to move through the main memory and your processor and so on and so forth. So this means that you're making lots of data moment. And probably as you've seen this slide before, the cost of data moment is much higher than doing simple operations in terms of the energy and the latency. So this means that there's a huge latency and the energy waste that is caused by this data moment. And one big reason is because the data analysis is performed far away from the data. And to solve this, we need to orchestrate algorithms and the architecture well so that we can handle the data and its computation well. So in general, if you are interested in this direction, of course, you can take a look at these materials that we make available on YouTube. So the links are already on the slides, so you can take a look at them. But to illustrate this on the genomic scale, I'll talk about this recent work from our group, which is GenPIP, which mainly focuses on the two steps in genome analysis and aims to reduce the data moment overhead. So as we covered before, this is particularly how the Nanopore sequencing looks like, right? So we have this Nanopore, we generate the electrical signals, right? These you'll have a bunch of electrical signals from many reads that you're sequencing. Then what you do, you store this raw sequencing data. This is electrical signal. This is not necessarily in a compressed form. It is essentially contains a larger amount of information than individual bases. But then next, what you do, you move this data to do some computation, specifically to translate these electrical signals into ACTGs using costly complex operations, maybe deep neural networks. So this is basically step by step what you do so that you can translate these electrical signals into the base called sequences. But then you store them. And then next you do another computation. You check, oh, this one is less accurate. You take a look at their accuracy scores, the quality scores that this base calling is giving you, so that you can prune some of the data that may reduce your accuracy, not necessarily impact your performance, but also more importantly, reduce your accuracy. So you prune some of those, and you'll continue with the remaining ones. You'll essentially store them again, and then you'll do further computation, maybe read mapping, right? So you'll look at reference genome and then see whether the read is unmapped or mapped and so on. This is also a costly process, and it also includes a bunch of data structures that we covered before. So what I'm trying to show is there are two main limitations in these approaches. One, this is not necessarily a limitation. As we know, there are multiple steps in genome analysis. But what we see is that there's a large data moment between multiple steps, right? Starting from the raw data generation up until the step that we get our answers. Second, there's a lot of wasted computation done on the data that is later discovered to be useless. For example, when you're doing your quality check, some of the reads will not be used in the later steps. So this means that you base code them for no reason. So it's a wasted computation, wasted energy, wasted time. So there are, of course, works that tries to elevate this, but I'll illustrate a bit more the first limitation before showing these works. So when you basically look at the amount of data moments that we're doing across steps, so we initially start with the raw signals, which this can be in the scale of terabytes, and then you base call them, then this data is compressed a bit, right? But now you have a few hundreds of gigabytes of data that you need to compute. So you move them, you process, again, some removed, then you do computation again, then later, what you end up having is basically this amount of data. But once you see basically the amount of data that you're moving across steps is basically, there's a huge data moment. But we don't stop from here because this data moment is not necessarily useful for us. Because imagine you process maybe 100% of your reads that you generate from base calling, but then some of them will be eliminated in the quality score checking, and some of them will be eliminated furthermore in the read mapping. So this means that you're also doing the data moment and also the computation unnecessarily because they are eliminated later on in the later steps. So how do you avoid some of these overheads, right? So one idea, let's try to then reduce data moment. And one idea is, hopefully everyone knows now, is the use of PIM, right? So you can use PIM to do some computation. Here, specifically, we do processing using memory to perform the dot product operations on a non-volatile based memory. So this can do the base calling for us because, as I told you earlier, this base calling includes a bunch of neural net operations. So we can also use content addressable memory to perform search, right? So this enables us to identify, perform maybe sequence matching. And we can do it again in a PIM fashion so that we can reduce the data moments as well. And this is useful in the read mapping step, right? So then this means that maybe we can reduce the data moment in between these steps, which will hopefully enable us to improve the performance of these genome analysis and reduce the energy. But then as you reduce the data moment in between these steps, ratio-wise, you're increasing the ratio of data moment in between other steps that you're not dealing with, right? And also, we recognize that there is essentially no prior work that tackles the unnecessary data moment in between these base calling and the read mapping. So we try to understand whether there's a motivation, whether there's an opportunity for us to tackle this particular problem, right? So our goal is to efficiently accelerate the entire genome analysis pipeline, starting from base calling up until maybe variant calling or read mapping, by minimizing the data moment and useless computation and moment, right? So we do this motivation analysis. Here on the x-axis, we will show different configurations. And on the y-axis, we have the normalized essential speed up. Here, essentially compared to the no accelerator solution. Here, what we assume is that we have a PIM solution for base calling and read mapping. When we have this, what we see is that we can achieve speed of up to 2.7x. Then we assume there is no data moment between these accelerators, because you can use PIM for base calling and read mapping, but whatever you generate using these steps, you'll need to move them basically to the later step, to, for example, from base calling to read mapping. So we assume that such a data moment does not exist. And when we assume such a case, we see that we can increase the performance by 6x. Then we further go ahead and assume there is no data moment and there are no useless computations, there are no useless reads that were unnecessarily base calling and read mapping. This is the ideal case. So when we look at this, what we see is that we can achieve 6x speed up compared to the case where we don't have an accelerator. So this is a motivation for us, right? This is how we start. And then we see, okay, so how can we essentially achieve or get close to this ideal case? So this is what we do, what we aim to do in GenPIP. So GenPIP is an in-memory accelerator for genome analysis that tightly integrates the base calling step and the read mapping step, and then enables them to communicate with each other so that we can reduce the unnecessary computation and as well as the data moment. For that, we have two key techniques. One, chunk-based pipeline. So we don't necessarily fully, for example, convert the raw electrical signal to its base code version and analyze it. What we do is we go chunk by chunk. We only base call a small portion of it and then analyze it. And then if it is necessary, we continue with the further chunk. Second is early rejection, meaning based on the analysis that we've done so far on the chunks, we try to assess whether we want to continue analyzing this particular read or not, right? If, for example, this read turns out to be, maybe it's going to be useless for us, we can stop at any point so that we reduce or minimize the unnecessary computations that otherwise would have been computed by base calling this read fully. By doing so, we achieve essentially significant speed ups and the energy reductions, which I'll come to it soon. So this is the chunk-based pipeline, as I described earlier. This is an illustration of it. So what you do in the usual base calling configuration is you base call the raw signals chunk by chunk. This is how the deep neural networks work in their structure. But then what you do is you base call them chunk by chunk. Then after your base calling is finished fully, what you do, you assemble these chunk reads, and then you perform your quality score checking, and then your read mapping, right? So usual step-by-step process. So what we instead do is, so we still base call the chunk of data, because this is what base callers take. Then what we do is, as soon as essentially we generate this data, we can actually start our computation, right? So we can sort of overlap also the latency, because we can let the second step of base calling also run, but in the meantime, we can also do the quality checking and read mapping for the amount of information that we generated so far. And we do this as long as we either decide to stop at any point or maybe not stop up until to the end of the raw signal, right? And by doing so, you first, of course, do some overlapping the latency, save some cycles, but also if you decide reject early, you even save even a large amount of cycles and energy. So this is on hardware how the GenPIP looks like. So what we do, we use the prior PIM accelerators for base calling and read mapping. So this particular work is a in-memory base caller, and this is in-memory read mapper. But what we also integrate is basically a mechanism that enables these two accelerators to communicate with each other and also to stop the process whenever this is needed. So there is basically data flow control and the decision making going on in between. Here we show some results. We start with the performance results. Again, this is a normalized speed up to the CPU base. And what we see is that by looking at both the bacterial and the human genome, on average, what we see is GenPIP provides a huge speed up compared to CPU, GPU, and also the optimistic PIM, where we assume there is no data movement between the accelerators. So essentially, these are critical for the speed up. So we look at also the energy reduction that we're getting. Similarly, GenPIP reduces the energy reduction because it doesn't only reduce the unnecessary or reduce the data movement, but it also reduces the unnecessary data movement and useless computation. By doing so, we can even improve the energy reduction compared to this optimistic PIM because it still does some useless computation over there. Mm-hmm.
Between the two accelerators.
Yes.
But this work still has data movement between these two accelerators. We are overlapping.
Exactly. And also, you, by being able to decide rejecting early, you're also sort of reducing your amount of cycles that would have been wasted otherwise. So that early rejection also sort of reduces. So it doesn't really minimize up to the zero part, but it reduces towards that goal.
And the decision on early rejection is taken during treatment.
Right, exactly.
After some set of treats have been done.
Exactly. Exactly.
Is splitting data into chunks does affect some downsides. For example, like usually analyze everything to get more accuracy.
Yeah, excellent question. Yes, there is actually. We do this analysis, empirical analysis. We see what is the effect of chunk size to the accuracy, and we decide actually our chunk size according to that analysis. Since you asked this, I'll also mention the chunk size related to the base colors. So that particular chunk size is also a bit tightly integrated how the base colors work. And this usually goes with essentially you need a larger chunk of raw signals to perform the base calling accurately. But what we observe is, based on our raw signal analysis works and so on, actually your analysis, the read mapping analysis, for example, doesn't need that amount of chunk. So you could use a smaller chunk and then still perform accurate analysis. But just because your base caller needs a larger chunk, you're basically limited with that. And then you rely on that and then do your analysis. So this is basically one motivation that we're also using against, then let's use raw signal analysis, right? Because we can even focus on the smaller chunk of data to perform accurate analysis. More questions? Okay, so this is GenPIP. So you can see basically the idea is beautiful, right? So maybe simple, but also beautiful. It provides the important benefits by simply letting two steps communicate between each other. Maybe there are more ideas that we can discover similarly in this direction. So this is swordfish, as I mentioned earlier. I won't go into the details of it, but here what we essentially mainly look at is, so you can use memory-centric devices to train or to perform the neural net operations, right? This is a common case in neuromorphic computing as well, and this is not specific to genome analysis, because relying on the Kirchhoff's law and also Ohm law, you can do summation and the multiplication, which is basically useful for you to do the neural net operations. And of course, the question is, then why not rely on this also for base calling in the genome analysis case, right? We can do it. But then the question is, these memory-centrics are not really the ideal devices. So you're not going to always get the exact answer that you want to get from these memory-centric devices. So we call them non-idealities. There is some noise in these devices. So the question is, assume you train your model using some accurate hardware, maybe CPUs and so on. Now you have a trained model, and then you offload this model on a memristor, maybe crossbar RRAM-based array, right? So the question is, will you be able to perform the inference at the same accuracy that you would be performing on a CPU? So the answer is usually no, because of these non-idealities. So the question is, can we understand these non-idealities? Can we understand this noise better? And then somehow inject this in the training phase of base calling, so that our model is aware of these non-idealities, so that you make it a bit more robust, such that you do your base calling more accurately, given such a noisy hardware. So this is basically the idea in Swordfish, and this was presented at Micro last year. So we also look at how we perform further analysis using computing in memory. Here, we try to classify, we try to perform food profiling, which is relatively a common case in agriculture. So you try to identify whether your food, for example, is poisoned or not, right? But this is not done very commonly because of the cost of the operation. So if you could reduce the cost a little bit, if you could make it faster, maybe we can improve the human health, right? So this is a step towards that direction. We also look at how we perform some dynamic programming operations completely using PIM. In this case, we look at essentially the use of a particular processing in memory device that is commercially available, and then look at how we essentially offload certain approximate matching algorithms that are used for alignment to that particular hardware, so that we can perform the alignment in a higher performance and reduced energy. So we've been looking at processing in memory, but the question is, can we even get closer to the data and then do the processing, do the process even closer to it, right? And if I'm asking this question, probably the answer is yes. To illustrate this a bit more, I'll talk about this GenStore paper, which essentially focuses on the in-storage processing for genome analysis. So you've been looking at different levels in memory hierarchy, right? So you have the storage system, main memory caches, and now then eventually you compute it somewhere, right? So it may be CPUs or accelerators. What usually happens is that we need to move this data from the storage system, maybe from your SSD to wherever you're doing the computation. And we know that the computation could be a bottleneck, right? If you are doing alignment, for example, this is computationally costly. But also there is a data movement overhead. You need to move this data from the storage system to the accelerators or to the CPU. And maybe if you look at this perspective initially without designing any accelerator or anything, you may see that, oh, the essential execution time is mainly bottlenecked by the computation, right? Then you may want to accelerate this rightfully, because why not, right? So you're going to get a huge speed up. But then as you improve the algorithms, right? Maybe you design heuristics to reduce the amount of computation, you design some accelerators, you design some filters, the amount of data that you're computing. So this means that the bottleneck will actually shift. Maybe you'll elevate the computational bottleneck. So this means that maybe you're not going to spend a lot of time in computing, but you'll spend a lot of time by moving the data, right? So now the goal is to minimize this. And we do this basically in GenStore by filtering the reads that sit in the SSD that do not require maybe costly computation in the later steps. Rather, maybe we can do some simple computations within the SSD so that we don't have to move them across in the many levels of the memory hierarchy, right? So that you reduce the data movement. So this means you filter some reads by doing some computation within the storage, and you just only send the ones that are needed to be sent for example, to your accelerator. And you can do this essentially this type of computation as long as your computation is relatively cheap because you don't have the basically necessary the complex the computational capabilities inside the SSD unless you're designing really something very special for a particular application. So this means that, for example, we can design simple accelerators, simple logic to identify the exact matching reads. So identifying exact matching is very simple, right? You can do even some XOR and then identify the exact matching. Or maybe some non-matching reads, meaning maybe this read is highly dissimilar to a reference genome that we're going to analyze. And then you can somehow identify this huge dissimilarity again quickly so that you don't send this data because you know that it won't map when you send it. So this is basically, these are the two steps that we're doing in genome analysis. But of course, there are challenges. One is read mapping workloads exhibit different behavior given different technologies, different genomes. So how you design a hardware that is flexible enough. Also, as I said, there are limited hardware resources. So how do you utilize these resources efficiently so that you do the computation that you need to do in the SSD in an effective way? So by addressing these challenges, we propose essentially a GenStore-enabled storage system that can do the computation, the necessary basic computation inside the SSD, and by doing so, we can reduce the computation overhead as well as the data movement overhead. By doing so, again, GenStore achieves a significant speed up and energy reduction. So yeah, this is basically one work that performs in-storage computing for genome analysis. I'll move on to a similar work. Again, this work was again led by Nika, similar to GenStore. So here in this work, we look at a different application, which is the metagenomics. Okay, so we want to, rather than doing essentially read mapping to a particular reference genome, we want to perform metagenomics in the storage. So Megis, to this end, we propose Megis. Megis is the first in-storage system for end-to-end metagenomic analysis. And the idea is somewhat similar to GenStore, but of course there are differences so that we can fit the application well. Essentially, the idea is we want to cooperate the in-storage processing for metagenomics between the SSD and also the host system. Because you don't necessarily always use the SSD to do all the computation, but you can rely on the host system, maybe your CPU, to do some of the computations. And then if you can communicate, if you can create a communication, create a nice communication, let's say, between these two systems, to the host system and the SSD, maybe you can still accelerate the entire application. So to remind you how metagenomics usually works, again, we start with this bunch of DNA. Then what we do is, in the first step, we extract k-mers or subsequences from your input read set. Then there is this particular large database that contains the information of many species, right? So this can be up to 100 terabytes of length. So what we do is we query these subsequences to this database to identify the presence and absence information for each potential organism. And then we calculate the abundance distance based on these matching. So this means that overall, when you look at it, there is some k-mer preparation step, right? So you extract k-mers from the DNA, you prepare the data, then you query the database, then you perform some intersection operations to generate this abundance estimation. So this is overall the set of maybe computations that we're doing. So knowing this, what we realize is that you don't necessarily have to use SSD for the entire operations in metagenomics. For example, you can process the input data, right, which is your reads. You can extract the k-mers. You can sort them because sorting is not necessarily cheap to do it in SSD. So you can do all these computations in your host system. You can prepare the data. Then once you prepare the data, you can send it to your Megis-enabled SSD so that it performs the rest of the computation. But then still there are challenges, right? Meaning, for example, you're going to access a huge database. but you don't want to randomly access this data. So somehow you want to optimize your accesses so that you minimize the randomness. So this means that, so how do you lay out the metagenomic database such that you minimize this random accesses? And how do you essentially communicate or arrange this communication between the host system and the SSD and so on, so that you overlap all these latencies and so on. And for intersection, we have a small accelerator over here. So once you fetch the necessary data from the large database within the SSD, you can directly do the computation right where the SSD is located, right inside the SSD, so that you don't have to send this huge data to main memory and then do the computation in the CPU and then send it back again. So you can do it right over here, and then your simple operations are performed where the data resides. So this is basically, so as you can see, this is not simply basically using the SSD, right? So this requires you to understand what are the limitations of SSD? What are the resources that you can use in SSD? What are the requirements that you need to take care of so that you deal with your data layout, right? And also, what are the other, basically, the hardware that you can rely on, right? So you can imagine a heterogeneous system here, right? It doesn't have to be a CPU over there. Maybe there is another operation that can benefit from the GPU. So how do you communicate these two and then perform the data flow efficiently so that you can hide certain latency? So this is basically, if you want to learn more about these, the Megis provides really good insights, basically, how you optimize all of these steps nicely. So when you look at then the results in terms of the speed up compared to the software baseline, here the software baseline is, we have a performance optimized software baseline, which is a fast metagenomics tool running on a CPU. There's an accuracy optimized baseline, again, a slow tool, but extremely accurate. And then we have Megis over there. So we look at two different SSD configurations. One is the cost optimized SSD. The second one is the performance optimized SSD, where you have a higher bandwidth. Looking at both, we see that Megis improves the performance quite substantially compared to, of course, the CPU baselines. So you can see, basically, by simply just reducing the data movement and doing the important operations quickly inside the SSD, you can simply perform much better than these CPU works. So we also look at the energy that we're consuming using the performance optimized, accuracy optimized baselines, and also looking at PIM. Here, what we see is, again, since you minimize the data movement, so essentially you reduce the energy consumption, because the PIM usually requires a higher power consumption than simply the SSD. So by essentially relying on the SSD and the computations inside the SSD, you can also reduce the overall the energy consumption compared to PIM as well. There are, of course, many other analyses, so if you're interested, you can take a look at the Megis paper. So yeah, this is Megis. If there are no questions, I'll move on with the GNASM. So this is relatively a different direction. This is still an hardware accelerator. Okay, then I'll move on. So one of the last works that I'll cover is GNASM. This is an hardware accelerator for accelerating approximate string matching, which is commonly used in genome analysis and which is found to be one of the most computationally intensive steps in genome analysis. So our goal is to accelerate this step. And this is basically the first approximate string matching acceleration framework for genome analysis. So we're saying framework because this is highly flexible to certain parameters. So it uses an approximate string matching algorithm called BiTAP, because it can perform this approximate string matching using simple and fast bitwise operations. So this is really suitable for simple, cheap hardware. But we don't stop there, because BiTAP has limitations. So we modify the algorithm as well. so that we provide the support that we need for alignment. So this is actually a first work that shows that on the algorithmic level as well, it's not just the hardware level, on the algorithmic level, it shows that BiTAP can do more what it can do, let's say. So I'll simplify it for now as is, but in the later slides, I'll explain what it means. So we co-design algorithm and hardware together, and also optimize the memory placement so that we can reduce the data moment in GNASM. So this is how the overall hardware looks like. So we mainly have two parts. One is the GNASM DC. This refers to the distance calculation DC. Here we literally directly use the BiTAP algorithm. So we accelerate the BiTAP algorithm here to perform the approximate string matching. But what is further needed in alignment is the approximate string matching tells you essentially the edit distance, which is basically the minimum number of operations that you need to do to make one sequence identical to another one, right? So this is good information for us if your goal is to check whether particular sequence is similar enough to another sequence. But if your goal is to identify what are the exact operations, right? Maybe the insertions, deletions, mismatches, and so on, to convert one sequence to another, then the BiTAP will not give you that, which is basically usually generated at the traceback step. So we modify the BiTAP algorithm so that it supports the traceback, meaning once you generate your once you identify your edit distance, what we further do is we reuse the information that we generated so far, so that we do traceback to identify the operations that gives us this minimum edit distance. So this is the TB part, the traceback part in GNASM, and this is again done by some simple operations I'll show next. So in the distance calculation part, we rely on a systolic array structure because the BiTAP algorithm is a simple bit vector, bitwise based. It includes some bitwise operations using bit vectors. So this means that you can have simple logics, and the output that you're generating in the previous time step or the previous step will directly be consumed in the next BiTAP step. So this means that you don't necessarily need to move this data back to maybe to far away from where this is stored, right? Or you can rather just place the next processing element right next to it so that you can directly consume it, because you know that you'll consume it directly in the next PE. So this is basically the systolic array structure that we're utilizing in GNASM, and this is the bitwise operations that we're doing for the distance calculation. So further, once we generate all our bit vectors to identify the minimum edit distance, we then iterate on these vectors quickly so that we identify the optimal alignment operations, again using some simple logic here. So this is the TB part in GNASM. So by doing so, meaning you accelerate the computation, yes, right? So you do the bitwise operation, you essentially pick a nice algorithm that is nicely suitable for hardware, but you don't stop there. You essentially efficiently design it on hardware. But again, you don't stop there. You understand the data moment between steps, so you minimize the data moment. And by essentially co-designing all of these nicely, what we show is we can achieve a substantial improvement in terms of the performance and reduction in the energy consumption. So basically, I'm explaining this in a high level so that I can at least tell you a little bit just about what we essentially look at, right? So we don't simply implement an algorithm on hardware. We understand the application, and we co-design the application algorithm and the hardware accordingly so that we will minimize the potential problems that we are likely to have if we would have simply implement that particular algorithm on a hardware. So this is basically the beauty of the hardware algorithm and software co-design. So if you're interested in learning more about GNASM, you can take a look at this talk video that was delivered by Damla, who is now at 10x Genomics. So we also further improve the Scrooge, further improve the GNASM algorithm by doing some optimizations and implement it on not just on the hardware, but also for CPUs and GPUs. So meaning that there is still like room for further improvement, and one can work on it to provide further performance and energy improvements. This is a talk video if you're interested. The next work that I'll cover is SAGRAM. This is mainly built on GNASM, but it looks at the read mapping problem at a different perspective by focusing on the sequence to graph mapping. So remember, as I showed you earlier, we mainly use a single reference genome, but there are certain caveats to it. So the question is, then can we use graphs? We know that we can use graphs. The next question is, what are the computational issues, and can we accelerate them? So SAGRAM focuses on these issues to accelerate the sequence to graph mapping. So this is essentially the problems with analyzing the reads using a linear reference genome. And if you use graph, you can annotate the potential variations inside the graph so that you can perform more accurate analysis. So sequence to graph is useful. And these are essentially the steps in the sequence to graph mapping. So this is mostly similar to regular linear reference genome mapping. So what is mainly different is basically the seeding. Of course, the graph construction part, you construct your graph. That is the different part, but also the seeding part. So the seeding is not necessarily now just a simple matching to a linear reference genome, but you need to match it to a graph. But then further on, how to identify basically the right path in the graph by performing the alignment and so on. So essentially, we look at this pipeline, and what we recognize is this particular alignment part is now more costly. So we know that it was already costly in the linear reference genome mapping because it includes many dynamic programming operations. But essentially, the nature of dynamic programming operation in the linear reference genome case, it looks at a certain number of cells to perform the dynamic computation. But when you move on to the graphs, the amount of cells that you need to compute in the dynamic programming manner, they increase. And also now there is no particular pattern in the cells that you're checking in the DP. So this means that you can almost look at anywhere in the DP table to do the alignment. So this makes the computation a little bit more complex, meaning it is now slower. So we try to accelerate the entire sequence to graph mapping on hardware. And when you look at it, it actually is mainly similar to GNASM. So what is different is we have this now min-seed part over there, which does not necessarily use all subsequences in your read. It rather samples. It performs some sampling in your read. So it reduces the amount of data that you're processing. So this requires, this is done by finding some minimizers. So this is a sampling technique that is commonly known. And it requires some sorting and so on so that you can identify some minimum, let's say, data, local minima in your data. And we do all of these basically, again, on hardware so that we identify these minimizers and so on. But then you use this sampled data to perform the mapping and the alignment. So what is different at this part? So this part mainly uses GNASM, but what is different is basically the hop queues. These hop queues essentially enables you to look at further back in your dynamic programming table so that you can perform the computations that the sequence to graph is requiring you to do so. So this is basically the part that we're mainly modifying in SAGRAM. And by doing so, we can perform sequence to graph mapping on hardware. So yeah, if you want to learn more about the inner operations and a little bit more details on the challenges, you can take a look at this talk that Damla gave at ISCA when she was presenting SAGRAM. So yeah, this is SAGRAM, and this is a very good point to stop, because in the next part of the lecture, I'll mainly cover some algorithmic optimizations rather than the hardware optimizations. Yes, we can take again another 15-minute break and be back at 3:15. But to fill the time a bit, I updated the slides, so now we have we have more slides. Yes, so that I can go faster again. But yeah, I guess we will finish a little bit early. I assume we can go early, hopefully. Okay, so the last slide was the SEGRAM. We covered some hardware acceleration works from our group for genome analysis. So I'll also now cover some other optimizations that we published so far, some of the papers we published so far in our group. For example, we asked the question, what happens basically when we have a new version of the reference genome, right? So these reference genomes are not constant. They are updated regularly, some minor updates every few months, some major updates every few years. So then the question is, essentially, do we move to this new reference genome or do we stay in the old reference genome? And you may answer this, of course, this question differently. If you didn't already do your analysis, of course, you can go ahead and use the new reference genome. But if you already did your analysis on the old reference genome, the question is always, is it worth, basically, is it worth redoing your entire analysis again on the new reference genome? Because you don't know whether using the new reference genome will be useful for your particular read set. Maybe the information is exactly the same, looking at the part in the reference genome that you want to analyze. Or maybe there are some important changes, but you're still not sure, right? So how can you decide this efficiently? And of course, people are also asking, when is it the right time to change the reference genome substantially? Because it usually improves the accuracy, but it comes at the cost of additional computations that you need to do, which is the entire read mapping from scratch, right? And you don't want to do this unnecessarily. This is what we aim in Airlift. We want to mitigate useless computations whenever the reference genomes are updated and whenever you want to move from the old reference genome to a new reference genome. So a key observation, as I already told you, reference genomes are updated. The key idea that we are proposing here, not basically the entire reference genome is going to be substantially updated. So there will be a large portion that will stay constant. It will stay unchanged. So our idea is, can we quickly recognize the differences between the old and new reference genome, and can we leverage this information when doing the read mapping? So this is the illustration of potential changes that we may have in the new reference genome. So assume this is your old reference genome and this is the new reference genome. And we label mainly 4 parts. The first part is the constant regions. So those are the regions that do not change at all. So they stay exactly the same in terms of their base content. So we also have updated regions. So those are the regions that have slight changes, maybe some single character change, a few character changes between the regions. There are some regions that we call retired regions that do not appear anymore in the new reference genome. So they are completely now gone. They are deleted. And there will be some regions that we call new regions that will be completely novel in the new reference genome. So those are basically the 4 complete changes that you can have between the old and the new reference genome. So the question essentially is, can we leverage basically this information? And what we do in Airlift is that we create a dictionary, right, that gives you some coordinate mapping. So assume your mapping was at this particular position. So you query this dictionary and then ask, what should I do? So basically, this is the question that you're asking. So if the dictionary tells you that, oh, actually, you're at a region, you're at a constant region. So what we can do is we can simply just update the mapping position and nothing else. And nothing else means, for example, the alignment information, right? Maybe the edit operations that we identify in the old reference genome. The reason for that is mainly because, again, this is a heuristic decision, but this particular region is not changed. It is constant. So this means that whatever the operations we already performed on the old reference genome, we should be able to transfer that information to the new reference genome. So only thing that we need to change are the positions, because these can move right and left depending on the retired and the new regions, right? But of course, the dictionary can also tell you, or maybe you have a read that is under this retired region. So then what it enables you is essentially tells you, actually, you need to redo the alignment. You need to redo the read mapping for this particular read that falls under that region. So for these reads, you do the read mapping from scratch, right? So then it basically sort of like a man in the middle, a guidance for you, so that you don't necessarily do the read mapping for all of the reads that was previously mapped to your old reference genome. Rather, you ask, oh, can I simply just move the position or not? And if not, you do your read mapping according. Then we do some further optimizations for the updated regions and so on. So this is basically purely an algorithmic optimization, and this reduces the useless computations that you would be doing when moving to a new reference genome. So as I said, we are also looking at the newer genome sequencing technologies. For example, the Nanopore sequencing technology is a special interest for us. And previously, in this paper led by dogma, we looked at basically what are the challenges, what are the computational steps in analyzing the Nanopore sequencing data, what are the challenges, what are the basically the potential future directions for it. We also directly work on this direction. Specifically, as I also mentioned earlier, we try to perform the analysis, the genome analysis, by eliminating the base calling step, meaning we want to directly use the raw sequencing data and then do the analysis directly using raw sequencing data without any translator in between. And so this is basically, this raw hash work enables that. And I'll give you some key idea and the key insights, the observations that we generate from this paper. So the key observation is that we're utilizing, and that was also utilized in the prior works, is there is noise in raw electrical signals, in the raw sequencing data. And the noise means when you're sequencing a particular DNA content, assume you are sequencing ACTG, right? You'll generate an electrical signal. And then when you're sequencing again, maybe at a different time using some other nanopore sequencing device, you'll generate another electrical signal. So the observation is there is noise in these electrical signals, and this noise will cause basically generating similar raw signals, not exactly the same electrical signals, but similar raw signals when you're sequencing identical DNA content. And since we already have this observation, meaning the raw signals will be similar to each other, they won't be exactly the same. Maybe we can identify the similarity by calculating some distance between them, right? So you can encode them as vectors, maybe a bunch of signal values in picoamperes, and then you perform a vector distance calculation on a hyperdimensional space. But essentially, this is a costly step. So because distance calculation using millions or billions of vectors in a large space, if you especially worked on KNNs, you will know that this is a costly process. Rather, what we want to do is we somehow generate hash values from these raw signals, such that we can just use these hash values to match them each other. So basically, if these raw signals are similar, we want to generate the same hash value and then find the similarity based on these fast hash matches instead of the distance calculation. But there are certain challenges to it. One challenge, of course, how do you generate the same hash value from the similar enough signals, right? So because usually, especially when you're using a low collision hash function, if your input is even slightly is different, the hash value that you're going to generate is going to be completely different. And of course, we want to accurately find similar regions, as few similar regions as possible. Meaning, we don't want to find hash matches in a million of regions, but only a few hash matches, and those hash matches will be accurate. And these are some overview of steps in when we're performing real-time mapping while the sequencing is happening in raw hash without base calling. So what we do essentially, we convert the reference genome into its signal representation. So we know that how or what each sequence should look like in terms of its signal. We convert them, the reference genome to signals. And we somehow do some purification and denoising and so on both the reference genome and also on the raw signals. But then further what we do is that to enable the matching of signals, we perform quantization. So this means that the quantization approach will perform some bucketing such that the close by values will be assigned to the same bucket so that you can generate the same hash values from the similar raw signals. And what we do is that once we generate the hash values from the reference genome signals, we store them as usual in the read mapping step in the hash tables to use them as index. And the hash values that we generate in real time from the raw sequencing data, we query these hash values in the hash table, and then we get some matching positions if they occur in the hash table. Then we perform the conventional chaining and mapping steps, and then we continuously ask question. Given this chaining information that we generated so far, the mapping information, we continuously ask whether we want to keep sequencing or we want to keep mapping this particular data or not. Maybe we're not still confident enough to tell, okay, we want to stop sequencing. So if that's the case, if the answer is yes, we get the next chunk of data. Remember, these are generated in real time. So this means that all this operation needs to be done in real time, meaning the computation throughput should at least match the data generation throughput. That's why it is important to do all of these operations quickly. But then if the answer is no, we can actually tell the sequencer to stop sequencing. So there are certain functionalities that can do that. And then at the middle, basically partially sequencing the data, you can stop sequencing the read. So this means that you can reduce the sequencing time and cost that would otherwise be impossible without the real-time analysis. So this is essentially raw hash. This is the first hash-based work that enables the analysis of large genomes such as the human genomes in real time without base calling. I see something in the chat. So there is a question in the chat, or you don't see. I'll read the question. So the question is, so in raw hash, you hash the bucket, not the signal that was placed in the bucket. That is correct. So there is actually one more additional step to it. After, so bucketing is really like reducing the resolution of the signal, right? Imagine you may have, I don't know, 5,000 signal values. And bucketing, it will just reduce its resolution, maybe, I don't know, 16 buckets or 32 buckets and so on. So this is like a lossy compression, right? But then it will enable us to assign the close by signal values into the same bucket, meaning the same value. We could use the value, whatever the value we assign for that bucket for matching. But then actually there is an additional challenge to it because a single signal represents a very short region in the DNA. So this means that you will actually find too many matches if you just try to match this particular bucket value. So what we further do is actually, I didn't show this, what we further do is we somehow pack multiple buckets into each other by preserving the order of the DNA so that we essentially increase the region that we're spanning with this bucketing and packing approach. And then after packing, we generate some hash value from these packed values. And then we use that hash value for matching. I hope it was clear. So if it isn't clear, just write in the chat again. I'll take a look at it soon. So this is raw hash. Okay. So this is raw hash. We further improve raw hash by performing the quantization a little bit more intelligently by doing some mapping decisions in machine learning fashion as well to make more robust decisions. So this is what we provide in RowHash2. We also have RowAlign. In RowAlign, our goal is actually perform signal alignment. So after mapping the signal, after identifying where the signal might be coming from in the reference genome, this is what RowHash provides. It will just tell you the position. The next step is actually performing the alignment, right? So given the position, given the region, can you do the alignment? So the challenge to it is the alignment is costly. So this is an additional step on top of mapping. But then you are limited by a certain throughput, right? Because you want to do the analysis real time. So the challenge is actually how you insert something additional to the pipeline, but still achieve a good throughput. And we show that actually we can do this because the alignment operation gives us extra confidence, which enables us to relax some of the other parameters that we've been using in RowHash, so that we reduce the computational cost of the RowHash part. And by doing so, we essentially balance the performance by still improving the accuracy thanks to this alignment. So once we have this alignment information, the next step would be how you utilize this alignment information, right? So this is a good question to ask, I would say. So this is RowAlign. We don't stop here. We also ask the question, can we enable new applications in raw signal analysis without performing base calling, right? Because currently the challenge is the raw signal analysis is limited also by the number of applications you can do. You can map your read and identify maybe the organism and so on, perform contamination analysis or do alignment, but then the question is, what do you do with this alignment information? Or basically, what are the other applications we can do? We want to achieve this because our bigger goal is to completely get rid of the need for the base calling and perform full downstream analysis fully using raw signals. And in RowSample, we try to tackle one part of this work, one part of this direction. Here, what we do is instead of mapping your raw signals to a reference genome, we rather map them to each other, because it may so happen that you don't know what the reference genome is. It may be unknown, maybe it's not generated, right? So it doesn't exist, or you just want to create the genome from scratch. And the usual approach to do it is to find overlaps between reads. And we essentially transfer this idea to raw signal space, and we find the overlaps between electrical signals so that we can construct genomes directly from electrical signals without base calling. And we show that we can actually construct a single piece of fragment that is half the genome length of E. coli. So here, to illustrate this in a more numeric way, so here the half the length is around 2.7 million. And as I showed you in the initial part of this, in the earlier part of this lecture, the E. coli genome is around 5 million bases. So here, this single piece is constructed by simply looking at the overlaps between raw signals, and the length is 2.7 million. The average read length that we have in this particular database is around 6,000 bases. So then this means that you, by just simply overlapping, you increase the sort of the synthetic read length by about 400 times. So this is one, of course, the benefit of overlapping and constructing the assembly. There are many other benefits that we're excited to basically tackle as future work. So as we approach to the end of the slides, I want to iterate a little bit some of the points that I've been saying. But actually before that, I'll illustrate what other directions we can follow using this overlap information, right? So one direction is we can now use this overlap information, maybe improve the base calling accuracy, right? So this is an overlap information. This is a useful information that you can feed to your base caller, and then maybe your base caller will be able to utilize this extra information to make the model more accurate by maybe still making it less complex, right? Now, because you have a more useful information. As I said earlier, we want to perform the full downstream analysis fully using raw signals without base calling. The other question is, can we cooperate the raw signal analysis with the base code sequencing analysis, right? So this is a bit goes back into this maybe GenPIP idea or the other filtering ideas. You don't blindly base code, right? Maybe you do some cheaper operation and then decide whether you want to base code or not, maybe using the source signal analysis, and then you do your base coding. And of course, there are many other directions that by keeping the hardware design and the underlying sequencing technology in mind. So I'll now conclude with some future opportunities in genome analysis. So what we see is the computing is still bottlenecked by the data moment, because, as I said, there are many steps in genome analysis. There are many applications, and essentially, depending on the application, the needs will change. So remember the pipeline that I showed you earlier. But what we see is essentially the things are happening in the industry. For example, one of the sequencing companies, the Illumina, which produces one of the solutions based on sequencing by synthesis technology. So this is basically commonly as a market leader in the field. So they now integrate this FPGA solutions inside their sequencers, so that you'll do your analysis while the sequencing is happening. And this is basically essentially their mention for hardware acceleration inside the sequencer. So biotech companies are already realizing the importance of this. So this platform is Dragon, and probably I was lazy enough to update this slide, but there has been a recent publication related to this a few days ago in Nature. There is actually further improvement. There's a huge speed up for the entire genome analysis to reduce the entire genome analysis latency, such that we can now we can generate the data and then perform the full analysis starting from read mapping to the variant calling, and then do them maybe even within few hours. So if you are interested, I won't be able to provide you the link here. Maybe I'll update the slides later on, but take a look at this paper. It's pretty interesting. Although you won't be able to find the source code, which is something to discuss, I guess. Anyways, essentially the tech companies are also looking at the important workloads, the important computations that are used, the important fields, right? For example, I've been mentioning the dynamic programming computation, right? So DPs are used for many workloads. And of course, these tech companies want to provide the fast support for these custom operations so that we can perform them quickly. And what they do is actually they look at the genome analysis. So once you provide this DP instructions, the specialized instructions, can we compute them much more quickly than we've been computing before? And they look at basically the genome analysis workload over there, and then they see a 7x speed up by simply providing the DP instruction support on these H100 GPUs. Biotech companies and the tech companies are also now collaborating. They are describing their requirement solutions, the biotech companies and the tech companies are trying to provide solutions in terms of either software or hardware to these particular requirements. So AI ML operations are also very commonly used in genomics, as well as in many other fields, as we've been seeing nowadays a lot in the field. So there is this Cerebrus recent wafer scale engine, right? So this is from this year actually. We've been using the older version for some time. So this basically is a huge wafer scale engine that includes trillions of transistors to perform the AI ML operations in a much quickly and in a larger scale. So this is their benchmarking, and they show that they provide basically the best solution, the fastest solution compared to their competitors. So I took this image a few months ago. Maybe this is already outdated. since the field is moving a lot faster now. So we also look at the processing in memory solutions. We collaborate with the companies that provide these products, the solutions. This is one example. Here, UpMem, you can buy these essentially modules and then perform your processing in memory computations using these UpMem DRAM devices. We are part of this BioPIM project where we have many partners. This is basically an EU-funded project where our goal is to essentially elevate the data moment issues in genome analysis so that we can achieve fast and energy efficient and scalable genome analysis. So if you're interested, again, in this direction, as you might already guess, there are many, many materials available. You can take a look at these talks, watch them, and learn more about this direction. We also organize workshops related to this direction. This workshop was organized last year jointly with ReCamp. ReCamp is a top conference in the bioinformatics field, where we invited speakers to talk about how they accelerate, how we should accelerate basically the genome analysis workloads by co-designing hardware and software. And the entire workshop is available on YouTube. We also provide schedule organized courses. We have this genomics course that we offer every semester, and we make the videos available on YouTube, so you can take a look at these as well. So to conclude, today we covered many recent ideas to accelerate genome analysis, and also to analyze the genomes in ways that were not possible before. For example, the analysis of raw signal analysis and the assembly from the raw signals. So essentially, enabling cost-effective, fast and accurate genome analysis is important, but also another important item here is portable. So portable analysis has different implications, right? So you want to achieve a low-power solution that perhaps has resource that is limited by the computation resources. So how do you do the operations in such resource-restrictive environment? And how do you perform your analysis in an accurate and energy power-efficient way, right? So these are some important requirements, some important directions to take a look at. So other question is, can we do even better? This is, of course, a very general question. And probably the answer is usually yes for certain things. But what I'm specifically referring to is, can we take a closer look at the sequencing process, the sequencing technologies, right? So we took a closer look at nanopore sequencing today. I've described a little bit how nanopore sequencing works. But actually, can we take even closer look at maybe even nanopore sequencers or other sequencing technologies for analyzing other types of biological data, for example, the proteins? So I believe many future opportunities exist, especially with the new sequencing technologies, and especially with the new applications and use cases. So with that, I'll finish the lecture. So if there are any questions, I can take them. Are there any questions on YouTube? Okay, then we can end the lecture today. Thanks everyone for attending.